Okay.
Okay. Welcome to Thinking Beyond. I just want to announce next month's Thinking Beyond, and then we can get started. And we have John Mitrofanis, and he is talking on unraveling the mystery of brain cell chit chat. Do they talk with light? And then, as just mentioned, if you have any questions during tonight's webinar, just submit them using the Q&A feature at the bottom of your screen, and we'll get to those at the end of the hour. So I will go ahead and turn it over to the Beyond Center Director and our moderator for the evening, Paul Davies.
Well, hello, everybody, and welcome back. Most of you will know how we organize these events. We have a guest speaker. You're going to hear from her in a moment. Then there'll be a little bit of conversation between our speaker and Sara Walker. And I might even chip in myself using my position as moderator. And then we'll go to Q&A from you, our faithful audience. So let me just say a few words about Kate Adamala. So she works at the University of Minnesota, and she has this grand title of McKnight Presidential Fellow. It sounds wonderful. She works on some pretty scary stuff, synthetic cell engineering. So she can make her own cells. They're not living. She makes her own artificial cells. So she's a synthetic biologist, and she can investigate all sorts of things with drug development and doing basic research and bioengineering, and cast important light on the origin of life, which I think is something we're all interested in. She's a co-founder of the synthetic cell therapeutics company SynLife, and she's a polymath fellow at the Geneva Center for Security Policy, which already makes me feel a bit insecure, and co-founder and coordinator of the International Synthetic Cell Consortium, Build a Cell. So with that introduction, I've forgotten the title, but anyway, Kate, over to you.
Thank you so much. Thanks for having me here. Thanks for introduction, Paul. My title today is something different than I usually do. Usually when I speak to audiences like that, I use a variation of a title that says life but not alive. And that's actually a title that you, Paul, assigned to me. I remember we were at a meeting at some point, and we were all told that we're giving talks at a title that Paul assigned us. And that was the title that I was assigned by you, and I loved it. So I've been using it ever since. But today I will talk about planting new trees of life and what good comes out of it. That's what I will mostly focus on. But also what could be the concerns, what could be possible reasons why we shouldn't be doing, at least shouldn't be doing some aspects of it. So let me start with why, because I mean, that's a perfectly valid question. Why would you want to make a new tree of life? If you think about biology, you probably think about the fact that modern biology is incredibly diverse. And if you look at the life around you, you will see that there's this incredible diversity of form and function in every living organism. Life has done everything, one might think. But if you've been trained as a chemist, or even if you're a biochemist, biologist with understanding of the molecular basis of life, you understand that there is really only one life form that we know of on Earth right now. All of life comes from the same last universal common ancestor, and all of life is biochemically the same. So when you really look at the tree of life through the eyes of a chemist, it really is all the same. All organisms use the same 22 proteinogenic amino acids and the same 5 nucleobases. The energy currency is the same nucleotide. There are teeny tiny variations of the genetic code table in some few exotic organisms, but mostly even the genetic code table. So the link between the DNA and proteins is set in stone all throughout the tree of life. So all of metabolism, from the point of view of a chemist, is really pretty boring. And I know I'm probably making a lot of biologists angry right now here saying that. But when you're trained as a chemist, life is not that interesting because there is so much more that's possible. And then the kicker is that even though, I just said it's kind of boring, but it's still a black box. All of those all of this diversity, this surface diversity of biology boils down to one and the same biochemistry, which we still don't understand. That's really funny to me, that of all the centuries that we've done biology so far, we still don't understand how a single cell works. We don't have a full chemical ingredient list of a living cell. We don't know exactly how every protein, every small molecule in a living cell, what does it do, where does it go, where does it come from? So in short, we don't have a full recipe of a cell. So it's chemically not that diverse, but on the structure function level, it's very poorly understood. And that's a really crappy place to be as a scientist, because you're thinking, okay, I want to understand everything. I want to be able to change it, to modulate it. And I can't if I don't understand every single element of it. And that's the business model of our field, the field of engineering synthetic cells. We want to build a simple cell from scratch so we can actually understand what we're doing. And the motto of our field is a pretty overused quote from a famous physicist, Richard Feynman. It's been used to death, but it's a great quote, so everyone uses it. What I cannot create, I do not understand. And that's a very essence of our field. If we cannot create life from scratch, we cannot really say we truly understand living organisms. So what really is a living organism? What is a living organism simplified? Well, it's really a biochemical machine. That's how we think about it. A lot of the people in the field of synthetic cell engineering are fake biologists like me. I'm not really a biologist. I'm pretending to be a biologist right now, but I'm really trained as a chemist. And we have a lot of chemists, we have a lot of engineers, and we think of life as biochemical machines. So we think of life as a collection of molecules that does certain functions, has certain form, and we can learn from it how it works and how to modulate it. So what really is a synthetic cell? Well, the basic element of a cell is a membrane. And the word cell was actually given to those structures that the first person who invented the microscope, he saw them under the microscope and he thought, oh, they look like the cells that monks live in. And so I'll call them cells. And that wall of a cell or compartment of a cell is a defining feature of a cell. So we keep it. When we make synthetic cells, we keep that structure. We keep a membrane that's not very different from a membrane of any other living organism, including your own membranes. So we make our membranes from lipids, and specifically phospholipids, which contain a head group. And that head group is polar, so it sticks out to the water. And then there is a tail that's non-polar or hydrophobic, and that goes inside the membrane. And that's how we create those membranes, by arranging those molecules spatially in presence of water. Then there's cholesterol. We love cholesterol. I know the doctors yell at us that cholesterol is bad, but if you're a biophysicist making membranes, you love cholesterol because it actually makes membranes more stable and stronger. So our membranes have lipids and cholesterol, just like your own membranes. And now when we have a membrane, we put channels in that membrane. And those channels are there to bring stuff from the outside, just like channels in natural cells, and then to remove waste from the inside of that synthetic cell. And the channels are not very smart most of the time. They're just holes in a membrane. Our favorite channel is called alpha chemolysin. You might have heard of that channel because it's a famous channel in origin of life studies, because it's a model channel that's used to kind of try to elucidate the origin of membrane transport in the origin of life processes. But it's also the channel that gave rise to nanopore sequencing. If you heard of those tiny little USB stick looking like DNA sequencers that you can sequence DNA, and as of recently, you can also sequence proteins using those tiny devices with a very tiny amount of reagents. That technology originated from alpha chemolysin. It's not alpha chemolysin that's being used in those pores anymore, but the original idea came from alpha chemolysin. And the idea actually came from an origin of life researcher, Dave Demer, who was studying the use of alpha chemolysin in the origins research. That kind of shows you the connection between the modern cutting edge synthetic biology technologies, the healthcare technologies, which I'll talk about in a moment, and this very foundational research that we're doing. So at any time during this talk, if you think what we're doing sounds pretty useless, there are actually applications down the road. You might just not see them right away. So the reason we have a membrane is to separate the inside, so the cell of the cell, from the outside. And that doesn't have to be done with a membrane. Now, most of what I do is with a membrane, and life uses membranes, but there are other ways of doing it. And one of the ways of doing it is, instead of using a lipid membrane, you could use this so-called phase separation phenomena. So you could make droplets. You could make oil droplets or water droplets in oil, and that allows you to encapsulate things just like a cell does. And they even look, those oil droplets or water droplets in oil even look like cells. This is a video from my postdoc Theo, who loves droplet microfluidics, and he thinks emulsion microfluidics is superior to liposomes. So I'm showing that video to show you that not everything that looks like a cell is actually a cell. These guys have no membrane, but they have individuality, they encapsulate metabolism, and they behave cell-like. But back to the membrane, because that's what I really like to work with. So we have a membrane that encapsulates a synthetic cell. Now, what's inside that membrane is the really fun stuff. It's the guts of this, so we call it the guts of synthetic cell, the cytoplasm of a synthetic cell. And so part of that cytoplasm is the genome. So the genome of synthetic cells contains DNA, just like all other genomes of all organisms on Earth. And how big is the genome of a synthetic cell? So just to put that in perspective, if you look at the amount of genes, humans have about 20,000 protein coding genes. So protein coding, that means that's a gene that actually encodes a protein. And E. coli has about 5,000 of them. Now, the simplest living cell, the simplest natural living cell, a Craig Venter created mycoplasma-based synthetic cell, has 500 genes. Our synthetic cells right now are barely visible on that scale because we have anywhere between 50 and 100 genes in our little genomes. So it just shows you the scale of complexity. What we're building is really, really simple. It's truly the simplest possible unit of lifelike behavior. And so when we say lifelike behavior, what do we actually do? Well, we make proteins. Most of life is making proteins. That's kind of the business model of life, is you take DNA and make it into proteins. That's how life works. That's what life does on really when you think about it on a biochemical level. And the way we do it is we do it outside of the cell. So to make proteins, you don't necessarily need a living cell. You can use this so-called in vitro translation system, which basically creates a translation system outside of a living cell. So we isolate all of the elements that normally make proteins in cells. So ribosomes, all the RNA, all the small molecules, all the other enzymes, and we put it together to reconstitute a translation system that now we can control because we reconstituted it from non-living components. And now, how does it look like? If you really look at it under the microscope, this is what it looks like. Not much to write home about. It's just a blob. Here are 3 different images of the same synthetic cell taken in my lab. And what you can see is the green is the metabolism of a synthetic cell. So that's a protein, a favorite protein of synthetic biologists, a green fluorescent protein. Blue is a dye that shows you where the DNA is. You can tell there's no chromosome. DNA is evenly distributed. And red is a lipid membrane stain. So it shows you that there is a membrane encapsulating DNA and protein production. So does it actually make a cell? Is it really a synthetic cell? Not yet. So what we want to do is we want to make a synthetic cell that has all the functions of a living cell and is fully controllable. So right now, that cell is not yet living. And I know some physicists might disagree with me, that might say that something that we do, which can undergo a complete cell cycle, is already living. Most biologists say this is not living. So that's where I stand to. There is more biologists than physicists. So if I choose to make one community angry, I choose to make physicists angry and keep biologists happy because they fund me. So I'm saying this is not yet living. But what it has is all the elements of life. They're well-defined components, all the small molecules, lipids, enzymes, DNA, genome, everything that makes a cell. Now, there is another way of making a synthetic cell. If you probably heard of the synthetic cell that's made by Craig Venter, it's a mycoplasma-based synthetic cell that it has a completely known genome, but it's not fully understood, and it's made out of a natural living mycoplasma bacteria. And so you can see that there are those kind of two different approaches to making a synthetic cell. This so-called top-down, where you take a cell and make it simpler, and then the bottom-up, where you take things that are dead and you try to make them back into a cell. We're hoping that one day both of those approaches will meet in the middle, and we will actually make a synthetic cell of complete defined composition that's undeniably living. That's the goal. We're not there yet. That's where we're going. Okay, so that sounds crazy, right? What I just told you is that we want to take non-living molecules, put them together into a lifelike system. You might think that this will never work. Well, I want to show you a quote from an article that was published in the New York Times over 100 years ago. New York Times, a very respected publication, 100 years ago when the press was doing a little better, it was even more respected publication. Someone published an article on the feasibility of building a flying machine heavier than air. So the year is 1903. We have balloons, we have dirigibles, but the common knowledge and very accepted in the engineering and scientific circles is that in order to fly, a machine has to be heavier than air. So it has to be filled with either a light gas or a hot air. And someone published this whole long article, and just one quote is shown here that says, well, a flying machine heavier than air will never be created, so you might as well not bother. Don't waste your time. And then 2 months later, the Wright brothers flew their first airplane, the first flying machine heavier than air. The reason I love this quote is that it shows you that if you tell scientists and engineers that something is impossible, we're just going to go ahead and do it to spite you. And that's kind of where the field of synthetic cell engineering is right now. People say it's impossible to create life from non-living components. And I tell you, wait a moment, maybe not 2 months, maybe we won't be done in 2 months, but we'll be done within a decade. We'll be able to say, okay, this is how you make a living organism from non-living components. So now the question is, why, if we do it, when we do it, what is it going to be good for? So here's some applications of the synthetic cell engineering that are already being developed. It will help us to understand how biology works, because back to Feynman, if I can build it, then I can truly understand it. We will be able to understand the living systems on the molecular level. We'll be able to make better tools for bioengineering. And that's already working. The most commonly used cloning technique, so the technique that every lab, every modern molecular biology lab uses to build their plasmids these days, is called Gibson ligation. And that was developed by Dan Gibson when he was working at the JCVI on the construction of the first minimal genome of the mycoplasma synthetic cell. That shows you this interplay between technology development and making actual functioning synthetic cells. So this technology development for bioengineering is already happening. Better diagnostics and better therapeutics. Synthetic cells have already been demonstrated to shrink tumors in living mouse model. That's where that technology is right now. It's not just proof of principle. We actually are in the animals right now. Soon we'll be moving on to clinical trials with some of the synthetic cell-based therapeutics. Metabolic engineering. Most metabolic engineering companies in the world right now use cell-free systems to do prototyping of their pathways because it's much cheaper, faster to do it. So the synthetic cell technologies are already used to build better bioengineering methods, better bioengineering solutions towards bioeconomy one day. Space exploration, we're using it to make drugs for long-term space exploration. Once we send Elon Musk to Mars, he's going to have to make medicine. And that can be done using lyophilized cell-free systems that we are developing right now. It helps us understand the origins of life, how life started by reconstituting those processes in the lab. It helps us develop new materials. With synthetic cells, we can make polymers that nature doesn't normally do. And that opens up a whole new branch of material sciences. And last but not least, we can actually build functioning biocomputers because synthetic cells don't have the noise of natural cells that normally prevent biocomputing systems from working reliably and at scale. So these are all the applications that are being developed already. And I want to show you one kind of an area that's very kind of a hot topic right now for very good reason, because it touches every aspect of your life. It really affects everything we're doing right now. And that is pretty much encompassing most of what's on this slide. That is the reliance of our economy on petrochemicals and oil. So when you think about it, you kind of irrationally know that our economy right now relies on oil. But most people, when they think oil, they mostly think energy. So you think that without oil, we wouldn't have electricity, we wouldn't have gasoline. But most people don't think about it. Without oil, we also wouldn't have pretty much anything else, because everything that surrounds you comes from petrochemicals, not just the obvious things that are made of plastic, but textiles, medicine, electronics, and food. There would be no way to run modern agriculture without petrochemically derived fertilizers. So the chemicals that we get from oil as if not more important than the energy we get from burning oil. Now, renewable energy, you say. We have a lot of different sources of renewable energy, but those sources, as the name implies, are sources of renewable energy. So if we finally manage to divorce our economy from oil and move on to the renewable energy sources, we will lose access to all of the petrochemicals. There is no way right now to replace the petrochemical industry, at least not at scale. How does that connect to synthetic cell engineering? Well, it does in a kind of a surprising way. So petrochemicals are made from formerly living organisms, now long dead. There are technologies that allow us to build those so-called bioreactors. So reactors that take current biomass, growing biomass, and turn it into chemicals. So that process called biomanufacturing takes what's green and grows in the field, and outcome chemicals that can replace petrochemicals. The thing is, though, there is no way to do that at scale right now. There is no way to do biomanufacturing at the scale that we require to replace all of the petrochemicals, because what sits inside that bioreactor is right now natural organisms, because that's all we've got. And natural organisms are not stupid. They're not going to make toxic petrochemicals just because you ask them nicely. And to do that, to make all those toxic petrochemicals that will replace all the other chemicals that we need right now, we need new ways of doing biomanufacturing. We need synthetic cells. That's where this kind of a synthetic cell biomanufacturing idea comes in, is that we can use synthetic cells instead of living cells for large-scale biomanufacturing. They're safer, faster, and cheaper to use than natural cells for those purposes. And so that shows you that there are practical applications to what we're doing, and it allows us to basically scale up biomanufacturing much faster than the current efforts with natural biology. So that's all good. What other cool thing can we do with it? Well, if we have a synthetic cell, we can digitize the information about it. And if I can digitize information about something, that means I can send it over long distances. And I can send it, for example, to my collaborator in Europe, and overnight he can recreate my experiment. Now, okay, so what? I can FedEx him a sample too, and that's going to be overnight as well. But what about places where FedEx doesn't deliver? And there are places, when we think about, for example, long-term space exploration, human habitation in space, there will be bioengineering needs that will arise during a long-term space mission that cannot be addressed by sample delivery because there will be none. So you need to have a biomanufacturing platform that can be used on demand. And synthetic cells are such a platform because we can lyophilize them. And now the question is, do they work under spaceflight conditions? Well, as of recently, we have an answer that they do. This is a video that I had to be up at 3 a.m. to watch that launch. So now I'm torturing everyone with it. But it's also cool because it's my own sample going into space. I mean, how cool is that? We sent a sample of a lyophilized synthetic cell system that was rehydrated on orbit and produced protein. And that protein produced measurable amounts of fluorescence. Now, then follow-up experiment, our collaborators sent a lyophilized sample to the ISS, but where a human astronaut rehydrated it and performed protein synthesis experiments that were then read, the results were read with an on-station plate reader, and that also worked. So we know that it works under spaceflight conditions. So those technologies can be used to explore, not only to send humans into space for long periods of time, to create drugs during long-term space missions, to create nutrients, but also to model conditions under which life could have evolved elsewhere in the solar system. Right now, if we talk about bringing a sample back from Mars or from Europa or Enceladus, it's really difficult to imagine what are we going to be looking for, because we haven't reconstituted those origins processes elsewhere. Now, with synthetic cells, we actually can, because we can reconstitute those processes within those foreign bodies. And that can teach us about how life started elsewhere in the universe, not just on Earth. Okay, so a lot of cool applications and a lot of potential for good, a lot of potential for benefits that can come out of this technology. Now, the title of this talk also promised to address concerns. So let me talk about few potential concerns that come out of this technology. So before that, let me introduce you to your left hand. Now, let me introduce you to your right hand. If you look at your hands, they're mirror images of each other. The same with your feet. Your feet are mirror images of each other. And that phenomena extends to all of biology. Every biological molecule can exist in two forms, as we call them enantiomers. So every biological molecule can exist as one or the other enantiomer. Not just proteins, but small molecules, DNA, all molecules can exist as this opposite or mirror image of each other. And that creates the possibility that if you put together all the normal enantiomers of molecules into a synthetic cell, you could also do it with the mirror images of every molecule, and then you could create this so-called mirror life. This is not a new idea. People have been talking about it for a few decades. But as of recently, we actually started pursuing it as a reality. We started doing research towards booting up a mirror life, building a mirror life from scratch. That was actually a title of the National Science Foundation grant that I had for a few years, booting up mirror life. We literally wanted to do it. And there were a lot of benefits that we were thinking about when we wanted to build a mirror life. But there's also concerns about mirror life that we now, as we're working with ecologists and immunologists, started to realize. So everything in biology gets eaten or infected by something else. So if you have a cell in the environment, no matter what that cell, what that organism is, it will have something that will eat it. It will have something that can infect it. Now, the problem with mirror life is that it will not get eaten or infected by anything. It will not be infected by viruses. It will not be infected by any pathogens. It will also not be eaten by any predators because of the mirror nature of that molecule. It will be essentially invisible to the immune system and to the predators in the environment. And that means even if it's the wimpiest cell possible, it will just keep amplifying, because that's what life does. So if we have a mirror cell, it will start making copies of each other. Even if it does it really, really slowly, there will be nothing to stop it, because nothing eats it, nothing infects it. And that creates a possible concern that it could be an uncontrollable organism that will spread throughout the ecosystem and will not be controlled or contained. And that's why 3 months ago, me and other researchers in this field, we started advocating for an international ban or moratorium on this particular area of synthetic cell engineering. So we say, do not make mirror life, because there's no safe way of doing it. And that's one example of a scientific community coming together and self-organizing to call on moratorium on our own work. Like, I was pursuing mirror life. That was one of the subjects of my research up until early last year, when we voluntarily stopped because we realized there is no safe way of doing that. And now we're calling for the rest of the world to join us. And speaking of the rest of the world, in addition to developing those safeguards against this one particular type of synthetic cell engineering, there are many types of synthetic cells that can be built safely, can be pursued safely. And this is the goal of the community that I helped start 6 years ago, this community called Build a Cell, which is a community of people that believe in engineering synthetic cells and engineering practical applications of synthetic cells. We have labs all over the world, are members of our community, and we believe that to build a cell from scratch, you need this international community really working together. We have in-person meetings. This is one of the strengths of our community, is that we actually come together twice a year for an in-person workshop. And we have people from all over the world coming together, sharing ideas, sharing unpublished ideas. This is what we believe kind of a new way of going about the scientific enterprise. Let's share what didn't work so others don't reproduce our mistakes. And we believe that in order to really build a cell from scratch, we have to unite the scientific enterprise. Everyone who's a synthetic biologist who believes in the practical applications of it has to join this global effort to make a synthetic cell and also to safeguard synthetic cell engineering. Because as the mirror cell example showed us, the only way to do something is to do it safely, and people have to come together and all agree on the same precautions and same kind of a safeguards and ways to do it. And that's, we hope, is the future, is that we can build a synthetic cell, develop all of those practical applications, beneficial applications, but also do it safely as international community. And that's all what I had for today. I want to say thank you. I didn't show really any actual results from my lab, but I showed some images. And most importantly, my work would not be possible without our funders and most importantly of all, the people in my lab. I want to thank all of those amazing people that were dumb enough to join my lab. When I started my lab, I say, I want to make life from scratch. And I'm in a biology department. People have actual living organisms to study. And here's me saying, I want to put molecules together into a cell-like system. And people were actually crazy enough to join our lab. People were crazy enough to fund us. And I will be forever grateful to all of them. And thanks to all of you for listening. And I'm really looking forward to the discussions.
Well, thank you, Kate. You've given us a lot to think about, and we've got a few questions that are drifting in. I'd like to hand over to Sara for a little bit, because she's also an expert on chirality or handedness of life. And as you mentioned, Kate,
Yes.
all life that we know has the same handedness, left-handed amino acids and right-handed sugars, but it could be the other way around. So Sara, would you like to take up that discussion? And on the way to answer Barbara Temple's question, well, how did that handedness arise in the first place?
Yeah. So actually I have a couple of questions about it, Kate, because I was actually really surprised when I saw the report. I haven't read through all of it, so I don't understand the structure of all the arguments, but the thing that really struck me was how strong it seemed to be that like all development in this area should stop. Because of these stated risks. And I don't see those risks as being anything different than what you see with synthetic cells more broadly. Just there seemed to be some sense that they were amplified, but this idea that life on Earth has no ability to interact with mirror life is, it's like a very black and white interpretation of the biochemistry, because there are enzymes from racemase activity, and there inverts chirality. And there's lots of examples of organisms using opposite orientation molecules.
Yes.
Not an entire life form though. So obviously it's different. So, and I think, running ecological models, which I did a long time ago, actually, on looking at computer life forms, like there are ecologically stable interaction dynamics. So, so I guess the question is about why mirror life is viewed as particularly bad in the space of synthetic cell technologies. And then the related question is, if we come down on this as being very detrimental, my other concern is that has downstream consequences of stifling other areas of research related to of life. And I'm sure you've thought about all of this, but just as a community member that's like scientifically interested, and doesn't have as much opportunity in my own work to think about the ethical considerations, which you guys have considered very deeply. I like those were the things that I was
Yeah.
deeply about.
So the reason we think this is really the mirror life has a unique place in our little warring hearts is because of two things. One is the immune system response. Because we don't have a mirror organism, we cannot say for sure, but the immunologists concluded that because of the specific molecular mechanisms of the immune system activation, mirror cells would likely not be detected by the acquired or innate immune system. And that would potentially create a possibility of a very promiscuous pathogen that would not care about the host identity. And because of the mirror biology, it would be difficult to treat. So we can treat it with antibiotics, but you cannot treat all the crops and wildlife with antibiotics. And so that's the one concern, is that mirror life could be this opportunistic, slow brewing, but nearly unstoppable infection. We don't know that for sure, but because the concerns were serious enough, we think we better not find out. This is one of those things that if we're wrong, then everyone's fine and we're safe. But if we're right and someone doesn't make a mirror cell anyway, then we're all screwed. And that's one thing that I would rather not find out. That's one of the reasons why we believe it's too risky to actually find out, because the only way to find out is to make a mirror cell. And then what if we're actually right? What if the immunologists are right and it is an infection that remains stealth to the immune system? Then we have this infection that can spread in the wild. So that's one. Yeah.
Okay. All right. Were you going to add more?
Yeah, the second argument was going to be lack of predation. There are racemases in many organisms, but they only act on molecules. If you're a predator, you recognize your prey or your food by surface receptors. And if those don't match, then you move on. And so the idea is that a mirror cell in the environment might remain stealth to the predators, and there are no viruses preying on it. There are no bacteriophages that would be able to enter and replicate in a mirror cell. So again, it's a speculation because we don't have such organism, but better safe than sorry. That's the reasoning.
Yeah, it's super interesting because when I was also looking at the report, this is not a scientific question. This is much more of a cultural question. There seems to be a culture emerging of worries about existential risk with very little scientific support for the risk, and then engaging in behaviors based on perceived threats that are actually stifling scientific creativity and also like AI, for example, is one where there's a lot of people that are saying there's an existential threat for this technology. And the existential threat is actually not stopping the progress on the technology, but what it is making people scared to actually understand what the technology is and then feeding into narratives that are actually very. So I'm just wondering about sort of a general cultural phenomena where we are sort of like I think there's the real ethical concerns and what you're articulating, but also what are the opportunities of actually being curious and understanding the scientific pursuit, and how is this affecting sort of the rest of the synthetic cell community? Because certainly what I wouldn't want is for that kind of idea of existential threats, hypothetical or not, to actually stifle the scientific research across the board. And so how are you thinking about this with synthetic cell stuff broadly?
So we're obviously thinking about it a lot, because that's kind of the whole business model of my lab is to make synthetic cells. And we as a community thought about a lot before publishing that paper. It took us almost a year of work before we decided that we have to go public with it. And we were convinced by the arguments that the research was moving towards creation of mirror life fast enough that it was actually a possibility. It was actually becoming a reality, and there was no way to stop it other than discussing the risks in public. And I'm not a bioethicist, but I would like to point people to an article that Lori Zoloth wrote in, I believe it was a New York Times, right after the mirror cell paper came out. Lori is a religious scholar, and she is it's basically her whole job is to think about the intersection of ethical constraints and scientific freedom. And she pointed out that there is no freedom. I mean, she said it much nicer than I can say it, but the take-home message was that there's no freedom if everyone's dead. And that what has to underlie thinking in those areas is if a concern is serious enough, and in this case, and that's the royal we of ecologists, immunologists on this project, believe that the concerns are serious enough, then we better be safe than sorry. This is one of those things that I'm, as a chemist, I wanted to make mirror cells. I didn't recognize those risks. They had to be explained to me. And once they were explained to me, now I see that the benefits that we were pursuing are not worth it. We can still pursue some of the benefits of mirror life. Many of us are still working on mirror biomolecules. We're working on a ribosome that's capable of translating mirror amino acids and to make mirror peptide drugs, and that's still fine. That's safe because that doesn't have the ability to self-replicate. But we draw the line at the mirror self-replication, and I realize that that can backfire to the rest of our community, which is why all of us on that paper and other people in the synthetic cell community right now are screaming from the rooftops that what we're doing is different than mirror cell. There is a very big difference between a natural stereochemistry synthetic cell that can be eaten, can be infected, can be digested, and a mirror cell that would not be capable of those things.
Could I
That's
just
kind of what we try.
chip in to this conversation? So, Kate, it wouldn't necessarily be easier to make mirror life than to make life as we know it. So why are people worried? Because we're very far from being able to make any form of life in the lab. So isn't it a bit premature to be worrying about making mirror life?
Not really. The state of the art in the field is right now that we have a complete cell cycle and we have cytoskeleton that's capable of budding. The contraction ring doesn't quite finish yet, but that's where we are, is we're really steps away from self-replication. And most people agree that once we achieved a cell cycle homeostasis and self-replication, then we have a system capable of Darwinian evolution. And by most definitions, that would be life. So most people in synthetic cell engineering community agrees that we're probably about a decade away from being able to put together non-living components into a lifelike system. And some unpublished data that's been floating in the community in the last year indicate that we might be even closer. So it really isn't science fiction anymore, which is why we decided this is time to speak up, because the science is moving really fast on this. And we will be able to make this crazy thing of building life from scratch, probably sooner than most people realize. And that's why, if we're going to stop that train, the time is now before we actually have a mirror cell.
All right, let me hit you with something else quite different. Making mirror life in the lab is only dangerous if it doesn't already exist in the environment. How do you know there isn't a shadow biosphere of mirror organisms that has achieved homeostasis? It has mirror viruses that keep it under control and so on. If it was just microbial, we probably wouldn't have noticed it.
That's a question to Steve Benner, not me. I don't know if there is or isn't a shadow biosphere. We looked. Some of us looked really, really hard, and we haven't found anything. That doesn't mean it doesn't exist.
Right.
I believe.
But do you agree that if then making mirror life in the lab is no more dangerous than making conventional life?
If one exists in nature, then yes, then there is no difference.
So you should go look.
So we that's again royal we, not me myself, but people have looked for shadow biosphere for a really long time. And I believe I had those conversations with Sara a few years ago, and we discussed the concept that one planet can only support one biosphere. And that would be an argument for why there isn't a shadow biosphere. I'm not an expert.
But I would have thought that that's probably true if you've got the same chirality. But the one exception to that might be if you had a mirror life. Anyway, we've got a lot of questions, and I don't want to. It's your show, Kate. So I'm going to move us on to the questions. We've got many questions from Barbara Temple, and she's a regular of ours, and so that's no surprise. And she does say, well, what could we do if there was a pandemic? And I think you've already explained, Kate, that not a lot, I think, at the moment.
So yes, mirror antibiotics would work, but
Right,
there is no way to treat the whole biosphere. So you could treat a human infection, but not a wildlife infection.
Right. And we've talked a little bit. Well, she says on a related note, what's the ethics of using a biological computer? Is it right to use something biological like it's a machine? Well, I guess you've explained it is a machine. I see. Could a biocomputer be conscious? Well, you might argue that you and I are biocomputers.
Yeah, exactly. Biocomputers are conscious all the time. They're sitting on Zoom right now.
Right. I'd like to move on to a question that we received earlier from Sean Hamilton. And this is going down the path that, well, maybe the mad scientist will actually go ahead and make a second biosphere on Earth, whether it's mirror or otherwise. It doesn't matter. The question is, we've now got two trees of life. Will they coexist? Will one eat the other? Will they collide? Will this be World War III at the microbial level?
That's a very good question. And I think that goes back to the question that you, Paul, asked just a second ago, can a shadow biosphere exist? If we have a mirror biosphere, we think it would collide because we would be competing for the same carbon sources. And the amount of organic carbon sources on this planet is limited. So the amount of resources is limited, and we would be competing for the same limited resources. I mean, life on a planet is essentially a zero-sum game because there's only limited amount of resources. And so if you have it, that's part of the reason why the ecologists were screaming at us to stop mirror life work, because if you have this organism in the environment that's competing for carbon sources and nothing eats it, nothing infects it, it will eventually outcompete the biosphere that has a higher death rate. And that's, again, that's just speculations. I don't have experimental data on it because we don't have a planet where two biospheres compete.
Right, so we have a question about the anthropic principle, which I'm going to deflect because we have had entire Thinking Beyond sessions on that, so we'll come back to that another day. Brian Smith is asking if you make synthetic cells and then colonies of cells, and would they evolve to multicellularity and so on? I guess that may take a long time, but
a while.
Could you just go beyond just cells?
I mean, it took what, couple billions of years on Earth?
Right, yes,
so...
but nature did not have the benefit of Kate Adamala and her lab.
My grad students have rather short PhD timelines. So, I mean, yes, the idea is that we will be able to evolve higher order structures. People already work on synthetic cells in a tissue-like assemblies. For example, if you're interested in that, look up Hagan Bailey. He's UK researcher that does work in this area. He uses synthetic cells and organizes them in 3D structures that really, if you squint right, look like tissues already. So it's possible to engineer higher order structures. I'm not a multicellular organism yet, but definitely a tissue-like assemblies from synthetic cells.
We've got people obviously a bit rattled about the whole question of regulation and international agreement. So, for example, Darren Dugan says, well, what about mirror life research in China? Are you confident that they're playing the same game as us? Do you know anything about that?
So the leader of the synthetic cell engineering community in China is one of the authors of our mirror life papers. So, and at least in public, everyone is saying they agree with us. What's not public, nobody knows.
Right. And now, whether it's mirror life or anything else, that once it's, if you can make it and it gets out, so to speak, are you going to be able to monitor the spread of this new life form?
Yes.
And aren't there as many dangers as with mirror life that it will gobble up all of the resources that the sort of life that we know and love needs.
So if it's life made out of normal, natural enantiomer DNA and proteins, we will be able to monitor it because we have tools for that. We know what to look for. The problem with mirror life is that we don't even have monitoring. We can develop monitoring for it. We can develop mirror polymerases, but the resources required for that, we're in a country that can barely run a vaccination program. So it's rather doubtful that we would be able to monitor for mirror life. Natural enantiomer synthetic cells can be monitored like any other organism in the environment by sequencing and by DNA sequencing and protein sequencing. So anything we build, we can monitor, look for.
So a little earlier, I made this quip that nature took a long time to evolve multicellularity because they didn't have the benefit of someone like you. And we have a question, and it always arises. So, and it picks up really on Feynman's quote. And so if you can actually build something, then you understand it. So maybe you made life from scratch in the lab, and people think, well, therefore it's not so hard for life to arise because Kate Adamala did it. They've done it in Minnesota. Life has appeared. How likely, does that mean it was pretty likely that Mother Nature, without the benefit of chemistry, is going to do it? I've always felt that what we would like is to explain the origin of life without an intelligent designer, and you are an intelligent designer. The fact that you can do it doesn't mean that it's easy to be of nature. We can't. People often ask me, well, what's the probability that life will pop up, over 1 billion years or something? You must get asked that all the time. What do you say to people who ask you that?
Yeah, there is a big difference between studying a mechanism and studying a probability. When we build life in the lab, and if we reconstitute, for example, conditions on Europa or early Mars, it will tell us how the biochemistry could look like under those conditions. So it will give us the mechanism. It will not tell us how likely it was to happen. Because, as you pointed out, for many purposes, I don't feel that intelligent, but for the purpose of arranging molecules, I am more intelligent than random chance. So I am the intelligent designer that tinkers with those molecules to make the origins processes happen. And so the fact that I can make life in the lab does not mean it means life is easy to make. That these things have to be uncoupled. We will be able to understand the mechanisms of the origin of life, the chemistry of the origin of life, but it will not tell us how likely it is for a given body with water and the right temperature to develop life.
Nevertheless, if you crack the problem and you're able to make life in one way, there may be many ways of making it, but you do that, I suppose at the very least you would be able to elucidate one pathway that goes from life to life.
Exactly.
So that would give you a better idea if you could find an appropriate geochemical setting. Better idea as to how likely you are to go down that pathway. But I still have the impression that you're quite a long way from that. Now, we have a couple of questions from Kamesh Narasimhan. I'm sorry if I've mispronounced that, but I'll read the question anyway too. Given that any synthetic cell would have to be engineered with biocontainment mechanisms, wouldn't mirror cell life be expected to be biocontained in many different ways, e.g. with mirror amino acids? That's question number one. Perhaps you could answer that first.
Yes, if we wanted to make a mirror cell, we would definitely build in many auxotrophies into it. But again, using a popular culture quote, life finds a way. We think that the concerns with the release of the mirror life are significant enough that we cannot rely on those biocontainment mechanisms, because life is known to evolve around pretty much any biocontainment mechanism we give it. And when it comes to physical containment, there's the Murphy's Law. If you make something idiot-proof, they will just make a better idiot. And that's the kind of a rationale in the biosafety and biosecurity community, is that if you don't want something to be released, the only way to prevent a release is to not have the thing exist. Because if something exists, sooner or later, it will find a way out, either by accident or by malicious intent. For example, you cannot safeguard against bad actors. So if someone made Mirror Life, even if I made it with the best of intentions, a bad actor can steal it from me. And if it exists, I cannot prevent that from happening. A war can get started and my lab can get bombed and the thing can get out. The only way to make sure something is fully contained is if it doesn't exist.
Okay, well, we're nearing the end of this. I'm going to skip over the second question and go to one by Dan Dalton, which is a very specific question about the DNA that you're using. Is it folded up when you insert it into these cells? That is in both an active and non-active cell, normal operation, repair and or reproduction. I may have garbled that a bit, but do you understand that?
Yes. So the question is whether the DNA is properly folded, properly packaged. Our DNA is small enough that it does not need packaging. Our synthetic cells have genomes that constitute of up to 10 relatively small plasmids. They're big by plasmid sizes. Molecular biologists will scream bloody murder if I tell them I work with 100 kilobase per size plasmids, but they're small by biological standards. So we do not need to package our DNA. It's always active. It's always unfolded. We don't have histones. We don't have any packaging for our DNA because it's too small.
Okay, I think we've maybe just got time for one more question. Maybe two. This is from Joshua Barry. Is there a concern that by abandoning this research, when you've said that you're pretty much doing that, it'll be conducted less responsibly by others? And there is always that problem that there may be a secret lab somewhere in a country that's not very friendly to us,
Yes.
where they think they might do it. Though quite why? Because it seems like if there's a threat to the biosphere, and this is a bit like a doomsday project.
Yes.
It will zap them just like it will zap us. You can't make
And
it
that
specific.
is the hope. The hope is that everyone recognizes that Mirror Life, if it was ever to be weaponized, it would be a doomsday type weapon. And so no state actor is going to touch it. And the resources you need right now to make a mirror cell are international community or a state actor level resources, not a single disgruntled scientist in a garage resources. So that's the hope, is that by abandoning this research worldwide, we will prevent the resources needed for it to actually happen from being directed towards it.
And very much what has to, I think, be the last question happens to come from one of my students in my class. This is Shrihitha Goskanda, and I'm sorry, Shrihitha, if I mispronounced that. Would studying mirror cells in space be an effective safety measure? And we often hear this in connection with bringing back rocks from Mars, putting them in a secure space facility, like the International Space Station. If the astronauts die, don't bring it back.
There is no foolproof container that would safeguard against bad actors. And that's the problem. If you make it on a space station, yes, it would be physically separated from Earth and safe, but you could imagine a bad actor smuggling it back to Earth. So if it exists, it can be brought back. That's the problem. Planetary protection is not perfect. All of those safeguards that NASA developed for sample return, they're not perfect. They do not safeguard against malicious intent, which is a sad but truth.
Well, we're pretty much out of time now, and there's still a few questions rolling in, but some of them are repeating things that we have talked about. So you've stirred up some deep emotions, a lot of controversy out there among our followers, and that's good. And I guess that we can direct anybody interested to your research papers. And have you written any popular articles or done any YouTube videos? If they just
yes, there's been quite a bit in the last 3 months. There's been a lot of popular press on Mirror Life. So if anyone is interested, you can read that, or you can read the 300-page technical report that we put out.
Right, I think I'll pass on the latter. But anyway, it just remains for me to thank you very much, Kate, for an
Thank you.
inspiring, thought-provoking presentation, and to Sarah for her commentary, and as usual to the people who follow us and send in their questions. Come back in 1 month for something completely different. What's going on in our brains? Could there be some shadow information processing there based on photons? We shall see. Very speculative. But anyway, see you all in 1 month, and that's all from me. I'll say good night.
Thanks so much. Goodbye.
Thank you, Kate.